All right, so there's three steps, three main steps in respiration, right? So does anyone remember the name of the first step? Basically all that we talked about on Monday. Yeah, glycolysis. So number one, you've got glycolysis happening. And remember, glyco means sugar or glucose. Lysis means split. So we're splitting sugar. That's the first part of respiration. And then the other two pieces, which we haven't talked about yet, we have the Krebs cycle, also called the citric acid cycle. We'll talk about that today. And then the last step is oxidative phosphorylation. Oh, I misspelled. What's going on here? Oh, I didn't. Oxidative, yeah, phosphorylation. So on your handouts, if you saw the free energy worksheet that you guys did, I made comments if you got some of the questions incorrect, and one of them was asking how ATP is made, or the process that makes ATP, and a lot of you guys said respiration, which is correct. So how I worded it is correct. I was looking for phosphorylation. So phosphorylation is where a phosphate group is added to ADP. Okay, so we still have to get to these, the Krebs cycle and oxidative phosphorylation, but we've covered glycolysis, which is a pretty complicated part of respiration. So what we did in glycolysis, I guess I need to erase, so there's enough room. We'll just do this. So we'll get to those. We haven't done them yet. So in glycolysis specifically, we started out with a glucose, right? Sorry, glucose. Glucose is C6H12O6. So this is the molecule, the molecular formula of glucose. That's our sugar. That's what we're splitting in glycolysis. Does anyone remember what that, at the very end of glycolysis, we have one molecule of glucose to start. What do we end with?
Is it two?
Mm-hmm. Two what?
It starts with a P.
Yeah. Good, that's good enough. Two pyruvates. So it's a molecule you haven't heard of and probably will never hear of anywhere else, but you need to know that it's the end of glycolysis. So you have two pyruvates, and I'm not even going to worry about giving you the molecular formula. You have two pyruvates at the end of glycolysis. So we go from one glucose, and the really important element that we're looking at through all of this, in addition to how many ATPs we're making, is the carbon, right? So we had 6 carbons here. We're basically splitting glucose in half, more or less, and we're going to get, these are 3 carbon molecules. So pyruvate has 3 carbons. So we've just cut those 6 carbons essentially in half, and we have 2 3-carbon molecules. Those are our pyruvates. That, in a big nutshell, is glycolysis. So we end up with 2 pyruvates. Some other really important aspects of what happens in glycolysis. Do you remember how many ATP we end up with total at the end of glycolysis?
36.
That's the end of all of respiration. So once we get through the whole thing, that is right. 2, yeah. So we have, let's put other outputs over here. We get 2 ATPs. remember there's that energy investment and the energy payoff phase. So we're actually putting in a little bit of ATP, but overall we're getting to ATP. That's what I really want you to remember. So just a little bit of ATP. So we get 36 eventually, but we only have 2 so far in respiration. And then that weird molecule that I want to talk a little bit more about now, and we'll talk more about today, NADH. So NADH is our electron shuttle. We have to move a bunch of electrons to get to basically phase 3 of respiration. So we're kind of collecting a bunch of electrons, moving them over so we can use them in that last step, oxidative phosphorylation. So NADH is what's doing that. So do you guys remember how many NADHs are made? Anyone? 2, yeah. So that's an easy way to remember it. So there's 2 ATP, there's 2 NADH. That's what you get out of glycolysis. 2 pyruvate, additionally, never thought about it that way. 2 pyruvate, 2 NADH, 2 ATP. You get a lot of 2s in glycolysis. So these are the outputs you want to know. You want to know you get pyruvate, and you want to know that we get 2 ATP and 2 NADH. Those are the really important pieces because that's what's, well, the ATP, obviously, we're trying to make energy here, so that's important. These 2 pieces, the pyruvate and the NADH, will come back up later. They're going to show up again, basically. So those are the 3 main outputs you want to know from the end of glycolysis. Okay. So that summarizes basically everything I said, not everything I said on Monday. There's a lot more detail in there that you'll want to know, but those are the main really, really important pieces that you'll want to know for glycolysis. Okay. I want to take a minute here to talk about, I'm going to dig into NADH a little bit more. I don't have any slides on this, but I'll tell you kind of the main pieces of what you need to know. Are we good here? Okay, just a minute. So NADH, like I said, when you see NADH, think it's an electron shuttle. It's shuttling electrons around. And how that happens is through something called reduction. And I think I mentioned this on Monday, kind of quickly and in passing. And I don't want to go into too much detail about oxidation reduction reactions, but when you'll see, and you saw on some of the diagrams, you have this NAD plus turning into NADH. That just means we're adding an electron in the form of a hydrogen. So like I said, hydrogen has 1 electron. So you can think of this, we're adding an electron onto NAD plus. If you add an electron, it gets a little more negative, so you don't have that plus anymore. And now it's neutral. So this, just think of this as an electron. That's our electron that we are transporting over to the last stage, the stage 3 of respiration. So we're going to make quite a few of these molecules. A couple of terms you need to be familiar with. So this is going to be a reduction reaction. When you add an electron onto a molecule, it reduces the charge because an electron is negative. So that's why it's called reduction. So NAD plus is reduced in order to form NADH. That just means you added an electron. So reduction, you're adding an electron to something. The opposite of that is called oxidation. So if we went from, and this will happen too in photosynthesis, if we go the other way, NADH, and we chop off that hydrogen, basically the electron, whoops, NAD plus. So we form NAD plus again. That's going to be oxidation. Oxidation and reduction are just the opposites of each other. So oxidation, we're losing an electron. So you'll see these terms when I'm talking about going from NAD plus to NADH or vice versa. So I definitely want you to be familiar with reduction and oxidation, but that's really all you need to know. We're adding an electron or we're taking one away. And then that will form either NADH or NAD plus. If we take NADH off of here, that's basically going to happen at the very end where we're transferring the electron through this, what we call an electron transport chain. So we're, like I said, carrying electrons as NADH. So this is our shuttle. Once we chop that off through an oxidation reaction, we'll end up with NAD plus, and the electrons will go through this really long chain that I'll show you. That's basically how we make a ton of ATP. Okay, at the very end of respiration. So that's oxidation reduction. Any questions about that? Okay. I think that's all I wanted to, yeah. Those are the main pieces I wanted to kind of review with you before we move on. Let's get this going. So this is where we ended on Monday. I was talking about fermentation, which is basically just glycolysis happening over and over again. It's not nearly as efficient as when there's oxygen present. This happens when there is no oxygen. Okay. We talked about that. I know I said there's 3 steps to respiration. Annoyingly, there's this like intermediate step, definitely an intermediate step between glycolysis and the Krebs cycle. I don't know why it's not just lumped in with glycolysis. I'm sure biochemists have a legitimate reason for it, but it's this weird transition step between moving from glycolysis into step 2 of respiration, the Krebs cycle. So I want to talk about that right now. It's only one reaction. It's fairly simple, but it's going to give us the molecule that will then go into the Krebs cycle. So it's called pyruvate oxidation. So we already know oxidation means an electron is being lost, right? So that's why I kind of wanted to talk about what the heck oxidation and reduction are. Okay, so location-wise, we know glycolysis was out in the cytoplasm. We're not in the mitochondrion yet, but with pyruvate oxidation, we're moving into the mitochondrion. So glycolysis is the only thing that happens outside the mitochondrion. Everything else happens inside of it. That's why we consider it the energy center. Okay, so here you can see glucose to pyruvate. That's our glycolysis reaction. I'm going you guys that way. In pyruvate oxidation, that pyruvate that we made in glycolysis is going to turn into something called acetyl-CoA, and I'll show you what that looks like. And then that acetyl-CoA is going to go into this thing, the Krebs cycle. So this diagram is a nice kind of flow chart, even though there's a lot going on. It's a nice flow chart to show you kind of what molecules, the really important molecules, are going into which step, kind of the beginning and the end of each of these steps. Yeah. So it occurs specifically in the matrix of the mitochondrion. That just means right in the middle. So mitochondria, if you remember, have 2 membranes, just like a cell. They have a basically double double membrane. The inner membrane is all enfolded, so that's what this is showing, this kind of light color here. That's the inner membrane. This is the outer membrane. The pyruvate oxidation step is happening in the middle of, like, inside of both of those membranes. So the matrix is the most middle part of the mitochondrion. So for you guys on Zoom here, we have the outer membrane of the mitochondrion and then the inner membrane, which has a lot of infoldings. That'll come into play. It seems like, why does it matter where it happens in the mitochondria? But it'll come into play as we get into sort of the end of respiration. So just remember, for now, pyruvate oxidation happens in the matrix of the mitochondria. Yeah, okay. So what's pyruvate oxidation? Sorry for all the words here. I wanted to break it down and make sure you guys had the main details in print. So what are we doing? We're moving from pyruvate to acetyl-CoA. You don't have to worry too much about what acetyl-CoA is. I'll tell you a little bit about it, but it's what's going to go on to the next step. So we need it. We need acetyl-CoA, which is just a different molecule. So like I just said, pyruvate is oxidized, hence the name of the step, pyruvate oxidation. So it's going to lose electrons. Those electrons are going to move to our electron shuttle. So we're basically stripping away electrons from pyruvate, adding them to NAD+, which will then become NADH when we have that electron added. And now we have another shuttle of electrons ready to go to the final stage of respiration. So we end glycolysis with 2 pyruvates, right? So we have 2 pyruvate molecules. That means each of them gets oxidized. So each one is gonna lose an electron. So we actually end up making 2 NADHs. And then the final product, acetyl-CoA, there's 2 of those as well. So this step and then getting into the Krebs cycle is where it gets a little confusing as far as the numbers of molecules, because now we had 1 glucose, which was easy to deal with. Now we have 2 of the molecules, and going into the Krebs cycle, we'll also have 2 of these acetyl-CoAs that we're dealing with. So you oftentimes, depending on how the diagram or how I'm explaining it, it might be based on 1 molecule or 2. So I'll be as clear as possible about that. Just remember that now we're dealing with 2 molecules because we split glucose in half. So we're making 2 NADH, 2 acetyl-CoA, and we actually have CO2 as a waste product here. So this is part of the carbon dioxide waste that happens, that is an output of respiration. If you think back to the big picture reaction of respiration, there's 6 CO2 that are eventually released. Here we get 2 of them. All right, so what the heck is acetyl-CoA? If we break it down, again, looking at the carbons, it's easy to kind of count the carbons as we go along. So we have pyruvate here. It had 3 carbons. 3 carbons. We are going to essentially take one of those carbons off, and it'll be released as CO2. So here we have the reaction happening. CO2 is coming off as a waste product. So we're losing a carbon from this system. It's not disappearing, it's going somewhere else, obviously, it's being released. But that leaves us with a 2 carbon molecule. So we have 2 carbons here, we let go of 1 as CO2. So that's the acetyl part, 2 carbons with some other stuff attached that we're not going to worry about. And I noted that here. There's only 2 carbons because 1 was lost in the CO2 molecule. And then we attach what's called coenzyme A. That's why it's acetyl-CoA. We talked about coenzymes, I think, and how they can help basically enzymes work a little bit better. So, or no, those were cofactors. Coenzyme A is just another molecule that they don't even basically define here. So Coenzyme A gets added, that's why it's acetyl-CoA. What you really want to know about acetyl-CoA, because it'll come up in the Krebs cycle, is that we have 2 carbons. So we went from 3 carbons to 2 carbons. That's a really important drop because we're trying to account for all the carbons as we slowly walk through this process. So we release CO2, and then here you can see NAD plus gets that electron that gets basically torn off of pyruvate, and it becomes NADH. So again, this is just 1 molecule of pyruvate. We have 2 of them. So this happens twice. For every 1 molecule of glucose. All right, and that's all you really need to know about pyruvate oxidation. We're going into the next step with only 2 carbons. Okay. So here's our accounting table. We did not make, so we know what we made in glycolysis, 2 total ATP, 2 NADH, and then pyruvate and water. In pyruvate oxidation, we don't make any energy, no ATP, but we do make 2 NADHs, which are important. Again, further down the road, we'll need them. So here's our totals. We've got 2 ATP so far, 4 NADHs, none of the other electron shuttle, that FADH2 that I haven't talked much about. Okay. And then the 2 acetyl-CoAs, that's what's going to go into the next step. 2 acetyl-CoA are going to enter the Krebs cycle. That's what's going to keep this process going. And that is the end of the first step and a half, I guess, if we consider pyruvate oxidation of respiration. Okay. So now we're going to head into the Krebs cycle. Citric acid cycle, Krebs cycle, whatever you want to call it, and oxidative phosphorylation. So these are steps 2 and 3. They're all in this one PowerPoint. Okay. So the Krebs cycle is actually 8 different reactions. You will not need to know all of them. I'm basically going to point out the aspects that you need to know, kind of at the very beginning and the very end, and then how many ATPs, NADHs, et cetera, are made. Okay, so the Krebs cycle also in the matrix of the mitochondrion. So we had pyruvate oxidation in that kind of most central part of the mitochondrion, the matrix, as well as the citric acid cycle happening there too. All right, so this is the cycle. The molecules that are circled in red are the ones I want you to know, like I said, right at the beginning and right at the end. So I'm going to talk about this a little bit. We'll talk about the NADHs and the ATPs as well. Okay.
Where does it start?
Up here, yeah. We're going to start up here with acetyl-CoA. So acetyl-CoA from the previous step we just talked about, pyruvate oxidation, that's that 2 carbon molecule. It's coming into the Krebs cycle. And it is a cycle, so it just kind of keeps going around. And we're going to look at how the carbons change on the next slide. So we have acetyl-CoA coming in. You want to know that that is the very first part of the Krebs cycle. You need acetyl-CoA in order to keep it going. And that's going to turn into citrate, which is why it's called the citric acid cycle also. So acetyl-CoA becomes citrate. basically what's happening here, acetyl-CoA is combining with kind of the end product of the previous cycle, oxaloacetate. And that'll make a little more sense when we look at the carbons in just a second. So oxaloacetate will come up, bind with acetyl-CoA, and you get citrate. Some important notes to keep in mind. So remember, we have one glucose molecule. If we start with one glucose molecule, at the end of pyruvate oxidation, we have two acetyl-CoAs, right? So this is happening twice for every molecule of glucose. This is where it gets a little, I would say, a little bit confusing. Just keep in mind, this is one cycle, but if we have two acetyl-CoAs coming in, it happens twice. And that means these numbers will be doubled as well, the NADHs, the ATPs. And I'll give you those totals on another slide. So yeah, a note here, know the circled molecules and then the outputs. So the outputs are going to be, I'll talk about them right now. So it doesn't matter, don't worry about where they happen on the cycle because I'm not requiring you to know all of these intermediate molecules that are occurring. So we're going to have down here, it's annoying, it says, I should cover that up. GTP, it's ATP, it's the same thing essentially. So this is our ATP being formed. So in one cycle, we get one ATP. Every time though, with one glucose molecule, we get two cycles, right? So we have two ATP. And we typically, when we talk about respiration, we're thinking about the whole process as starting with one glucose molecule. So that's why I'm doubling these now. So we'll have two ATPs. We'll have 6 NADHs. So they are formed at a few different spots here. So in one cycle, we'll get 3, 2 cycles, we get 6. And now we'll have this other electron carrier as well, electron shuttle, FADH2. You can think of it essentially. It has the same purpose as NADH. So it's carrying electrons on to the very last step of respiration. And then within each cycle, we have 2 CO2s released, which means for one glucose molecule, 2 cycles, we get 4 CO2s. And the CO2 is just a waste product. Okay. So on the next slide, I think it's the next slide. Yeah. I want to look at the number of carbons. This might help explain a little bit of it. Okay, so same cycle. The acetyl-CoA is coming in, and it even says 2 carbons right there, right? So acetyl-CoA is coming in from pyruvate oxidation. It's coming into the cycle. Oxaloacetate is at the end of the previous cycle, so it's ready to basically keep going. Oxaloacetate has 4 carbons. Acetyl-CoA has 2 carbons. When they come together, when they bond at the very beginning of the Krebs cycle, you end up with a 6-carbon molecule. 4 plus 2. Okay, so we have 6 carbons in the next step, and then we're losing a CO2, so we're down to 5 carbons. And then we lose another CO2, and we're down to a 4-carbon molecule. So by losing those 2 CO2s, we end up back at oxaloacetate with 4 carbons. That's really critical. Otherwise, if you don't lose these CO2s, you're constantly adding more and more carbons to this cycle, and it would basically break down. Because you always have 2 new carbons coming in with every cycle in acetyl-CoA. And again, this occurs 2 times. Yeah. Okay. So we know the cycle occurs twice for every glucose molecule. Does that make sense? Okay. You have to reduce the number of carbons if you constantly have new carbons coming in order to keep the cycle the same. So that's why we sort of have to get rid of these CO2s. It's helping us bring the number of carbons back down to 4 so that when acetyl-CoA comes in, we can make a 6-carbon molecule citrate. All right. Oh, so here I have it basically written out. If it's helpful for you to see it in text, I have these different parts that you need to know basically written out in bullet points. So a review, we have acetyl-CoA coming in. It has 2 carbons. It's going to combine with the 4-carbon oxaloacetate, so we get the 6-carbon citrate. That acetyl-CoA is coming from that intermediate step, pyruvate oxidation. And then the oxaloacetate we just get from that ongoing cycle. We always, towards the end of one cycle, oxaloacetate is that. So that's coming from basically from within the cycle itself. Okay. I don't want to go too fast if you guys are still writing. So like I said, there's a lot more, obviously, that goes into the citric acid cycle. But if you understand those, essentially those 3 molecules, acetyl-CoA, citrate, and oxaloacetate, and how they combine to form the molecules with different numbers of carbons, I think that's plenty of information for an intro bio course. And then don't forget about the other really important outputs, the NADH, I might have this on the next slide. The NADHs, the ATPs, the FADH2. Because that's really the goal of all of this. We're trying to make NADHs, we're trying to make ATP. Okay, oh, let me just pull all these up. All right, so we're counting again, more numbers. Okay, we already know this. How many ATPs, NADHs, et cetera, glycolysis made, pyruvate oxidation, the intermediate step, just got 2 NADHs from it. And then the citric acid cycle, I don't know why I animated it quite so much. The citric acid cycle, here's the totals for that. So remember, this is all for 1 molecule of glucose. So this whole, every time I show you this chart, it's assuming we're starting with 1 molecule of glucose. So we got 2 ATPs from citric acid cycle. We got 6 NADHs, 2 FADH2s. We released 4 carbon dioxides as waste. And the other kind of output is that we're just continuing to regenerate that oxaloacetate, that 4-carbon molecule that needs to combine with acetyl-CoA at the end/beginning of each cycle. So the totals for these 3 molecules, so far we have 4 ATP, we've got 10 NADH and 2 FADH2s. So we've got a good number of electron shuttles already. We have 12 electron shuttles. We only have 4 ATP. We're eventually going to make 36 total, so we still need to make 32 more. And we only have 1 step left to do all of that. So we'll make 32 ATP in the very last step of respiration. Any questions about any of those numbers? Okay. just remember when you're going back and studying this and you're looking at that citric acid cycle, that's for essentially half a molecule of glucose. It happens twice. So that can get a little confusing when you're thinking about the citric acid cycle and you're seeing the totals here. But if you look at that cycle, it has half of these, right? Because I'm just showing one cycle. All right, on to the final step, the most exciting step. This is where we make all of our, not all, most of our ATP. And it's the final step of respiration, so that's exciting in and of itself. All right, oxidative phosphorylation. Oh, I started talking about this, I think, and then I got off track. But the free energy worksheets that you guys did, yeah, I did mention this. Phosphorylation is basically the process by which we're adding a phosphate to something, right? And oxidative phosphorylation, there's substrate level and there's oxidative. So substrate level is what we've been doing so far in respiration. It requires enzymes to basically make ATP. Oxidative phosphorylation is the much more efficient way to make ATP. So that's why we get a lot here at the end, because we're using just oxidative phosphorylation. All right, now this diagram is complete. So over here we have the last step, oxidative phosphorylation. This now, so we were in the matrix of the mitochondrion with these two steps. Now we are specifically, it's a very specific area of the mitochondrion where this happens. We're working on the inner membrane of the mitochondrion. So this, well, you can't even really see it, but this membrane that's folding over and over basically on itself, that inner membrane, is where oxidative phosphorylation is happening. And what I mean by that is essentially molecules, back to membrane transport like we talked about in the last unit, molecules are being transferred across this inner membrane, back and forth. And that's what's essentially creating a lot of the, well, is creating the ATP. And I'll show you how that's done. But the inner membrane of the mitochondria is where all of this happens. Okay, and there's kind of two pieces to oxidative phosphorylation. I'll talk about the electron transport chain, which you guys may have heard of in maybe a high school biology class, and then something called chemiosmosis. For the electron transport chain, that's why we need these NADHs and FADH2s. They're bringing all of those electrons from these previous stages over to the electron transport chain. Oh, yeah, here, electrons. It's already on the diagram. Okay. Oh, yeah. So here's a little bit more detail on that inner membrane. So yeah, the two parts of oxidative phosphorylation, we have the ETC or electron transport chain and chemiosmosis. And I'll talk about what each of those is in just a minute. We're in the inner membrane of the mitochondria, like I just mentioned, and we're transporting things across the membrane. That means we're transporting them between the matrix of the mitochondria, which is the innermost piece, so the innermost section, I guess, of the mitochondrion, inside both of the membranes. So in here, yeah, this would be the matrix, the innermost part. And then the intermembrane space. So inner membrane, INNER, versus intermembrane, INTER membrane space can get very confusing. So this infolded piece is the inner, INNER membrane. The space between that membrane and the outer membrane is called the intermembrane space, INTER. As if we need to make it more confusing. It sounds very similar, but intermembrane means in between two membranes. So we're moving stuff across this inner, INNER membrane, back and forth. That's the whole That's basically, yeah, what we're doing in this last step. Okay, so the electron transport chain we'll talk about first, and then chemiosmosis. The electron transport chain, we have those electron shuttles, the NADH and the FADH2, they're holding onto electrons, they're bringing them over to the inner membrane of the mitochondria, and then they're basically going to release them. When they release them, the electrons are passed along a series of protein carriers. So this should look, these protein carriers, I know there's a lot going on here, but here we have that membrane, the bilayer. So you still have a phospholipid bilayer membrane, just like a cellular membrane. And you have these proteins implanted in it. So they are going to play an integral part in this. So the electrons are basically passed along this kind of pathway using those protein carriers. Electrons carry a lot of energy. So negatively charged molecules, atoms, subatomic particles, whatever, typically have a lot of energy. So when you see a negative charge, think, oh, that probably has a good amount of energy. So electrons are no exception to that. They have energy. So as they're passed along this pathway, so here, this yellow arrow is showing the general pathway that the electrons are passed along. You don't have to worry about what any of these individual proteins are, like Q and 3 and site C. Don't worry about that. Just know it's passed along these protein carriers, and then that's going to release energy. Every time they're passed from one to the next, it releases a little bit of energy. That's not the ATP we're making yet. It's just releasing energy to allow for something else to happen. Okay. So we have, what was I going to say on this slide? Okay. Yeah. Oops. So the energy that's released by these electrons are getting passed along this electron transport chain. That allows for the hydrogen ions. So there's H+ ions. There's a lot of them in the mitochondrial matrix. They're getting pumped to that space in between the two membranes. So this is basically the ion that we want to move across the membrane are the hydrogen ions. So from the matrix, from that innermost part of the mitochondrion to the intermembrane space. The electrons, the energy from those electrons then being passed along this pathway are what's allowing hydrogen ions to go out to this intermembrane space. So what the cell is doing, what the mitochondrion is doing at this point, is basically building up a really high concentration of hydrogen ions out here in the intermembrane space. So pushing basically as many hydrogen ions as it can with the energy in the electrons out into the intermembrane space. And that comes into play in the next step. So we're building this really high concentration. And if you remember, if we have a high concentration of a certain particle in one area, by simple diffusion, it's going to want to come back to where there's fewer of them, right? So that's what's going to happen in the next step. So if we build up this really high concentration in the intermembrane space, these hydrogen ions naturally are going to want to diffuse down their concentration gradient, which means they'll want to come back in. That's where we actually make all the ATP. That's the next step. Okay. So before we get into that, a really important aspect and why oxygen is needed for respiration, why we need oxygen to actually fuel ourselves in each individual cell, is because oxygen is going to be what we call the final electron acceptor. I have bolded this. It is very important. The final electron acceptor in the electron transport chain is going to be oxygen. So these electrons are being passed along, right, along this nice little chain. They're releasing energy. That means hydrogen ions are going to the intramembrane space. But what happens at the end? These electrons have to go somewhere. They don't just disappear. We can't just lose electrons. So they end up on oxygen, basically. Don't worry about the 2.5, but we end up with electrons being added to oxygen. That's why oxygen is the final electron acceptor. Do you guys have any idea why oxygen is a really good final electron acceptor? Think back to water. When we were talking about water, you have the 2 H's and the O, and it's a polar molecule, right? So which of those are the H's or the O holding on to the electrons a little bit closer? Or which one is more greedy for the electrons?
Maybe because it's closer to a valence electron.
Wait, sorry, which one?
It's more the O because it's have more shells on it, so it was.
Yeah, exactly. It's really close to having a full outer shell, right? So oxygen is really highly electronegative, which means it wants electrons really badly. So it's greedy for electrons. That makes it a really good final electron acceptor because those electrons aren't going to end up somewhere else on some other molecule. Oxygen is going to grab them and hang on to them. So it's a really good final electron acceptor because it is, you would say it's very electronegative. But that is because the valence shell is really close to being full. Okay, and then we'll stop there and talk about what these hydrogen ions do in that last step on Friday. Don't forget to take the quiz. I forgot to mention that at the beginning. The quiz is up. Take it by 11:59 tonight. And we'll go over that on Friday too.